Hello बच्चों, welcome to SPSVERSITY. Today we are going to discuss about an interesting topic that is in plant physiology, that is photosynthesis in higher plants. So photosynthesis is an important function in plants where it prepares its own food materials. So that shows it is autotrophic nutrition. The process behind autotrophic nutrition is photosynthesis. Okay. When you come to importance of photosynthesis, so it is the primary source for all food on the earth. Whether you take carbohydrates, proteins, whatever it is the primary source for all food substances on the earth. And it is responsible for release of oxygen into atmosphere. So oxygen plays an important role in aerobic respiration. So oxygen is required for aerobic respiration, and that is generated from photosynthesis. So photosynthesis is a very important process for primarily for the food purpose as well as for oxygen purpose. So when you come to the experiments on photosynthesis, when you come to historical perspectives, many people conducted experiments to know about photosynthesis. So what are the factors affecting photosynthesis? So when you come to this experiments, उसका नाम क्या है कि जोसेफ प्रिस्टली. He has done an experiment by taking a bell jar. So he has taken one bell jar, and inside bell jar he kept one candle and one mouse. After some time, what happens is the mouse died and the candle turns off. So the reason is whatever oxygen is present here, it is used by these two. Okay. In the second step of the experiment, what he has done is, along with candle, mouse, he has kept a plant. Even after some time, there is candle is burning and the mouse is alive. So what is the difference between these two is a plant is kept here. So what this plant will do? What actually happening inside the bell jar? Okay. So if you take a burning candle and animal, both require air. Okay. And they release carbon dioxide. But when you kept a plant, he has kept a mint plant. So from this mint plant, oxygen is released, which will keep the candle burning and the mouse alive. Okay. So what is the conclusion by Joseph Priestley? The conclusion is plants restore the air. So restore the air with whatever is required for breathing of animals and burning of candles. That is indirectly It will take carbon dioxide and release oxygen. It will purify the air. So this is the conclusion given by Joseph Priestley. So plants restore the air, whatever it is required for breathing animals and burning candles, which are released from these two. Okay. And the next person, Jan Ingenhousz. Okay. He has showed that only the green parts of the plants will release oxygen. So green parts of the plants means leaves, younger stems, these all are green parts of those which have chloroplast. Okay. To prove this, only green parts of the plants will release oxygen. So he has done an experiment on an aquatic plant. Aquatic plant means which lives in water. Okay. For his experiment, he has took one aquatic plant. Okay. And he has kept this aquatic plant in a funnel, and this funnel is kept in a beaker of water. And this is totally set up, is kept under bright sunlight. So after that, he has observed small bubbles were formed around the green parts. But whereas non-green parts, there is no bubbles like this. So what are these small bubbles? The bubbles are mostly of oxygen, which is released only from the green parts of the plants. Okay. So what Jan Ingenhousz has proved is only green parts of the plants will release oxygen. The reason for this is the green parts means they have chloroplast, which will perform photosynthesis. Okay. After Ingenhousz, next person, Julius van Sachs. Okay. He has proved the production of glucose when plant grows. Later on, he has showed that the plants will have the green substance, that is the pigment chlorophyll, which is present inside chloroplast. So chloroplast is the cell organelle. Inside the chloroplast, there are pigments, that is the green pigment, that is chlorophyll. So chlorophyll is a pigment and chloroplast is a cell organ. Okay. And what he has observed is the green parts in the plants, when you observe green parts of the plants where glucose is made. And this glucose is stored in the form of a polysaccharide, that is starch. That is, during photosynthesis, glucose is produced, and this glucose is stored in the form of starch, and this happens inside the chloroplast where the pigment's chlorophyll is present. Where the pigment chlorophyll is present. So this is the what Julius van Sachs has proved. Okay. So till now we have studied about Joseph Priestley, Ingenhousz, Julius van Sachs. Okay. Joseph Priestley proved that plants will purify the air. Ingenhousz proved that green parts of the plants will release oxygen. Julius van Sachs has proved green parts of the plants during growth they will produce glucose. Okay. बाद में एंगलमैन, T.W. एंगलमैन, So what he has done? So he has done his experiments on a green algae. The green algae का नाम cladophora. Okay. So he has used green algae which is placed in aerobic bacteria, in the suspension of aerobic bacteria. So what is the purpose of keeping this bacteria? The bacteria will be present where the sites of where oxygen is evolved. So he has taken a green algae which is kept in suspension of aerobic bacteria, and he has used a prism which will split the light into spectral components, that means different colors, WIPGR. Okay. So what he has observed is bacteria accumulated in the region of blue and red light of spectrum, that is visible light. Okay. So here the bacterial accumulation, it shows that in that region only oxygen is released. Majority of oxygen is released. Okay. Thus he has explained the action spectrum of photosynthesis. What is meant by action spectrum? The action spectrum means the rate of photosynthesis at a particular range of light. Okay. In all the light components, the rate of photosynthesis will not be seen. Okay. So here you can see this. This is prism. So when white light is passed through this prism, it will split into a visible range of light, that is WIPGR. Okay. And you can see here the most of the aerobic bacteria are towards blue and red regions. So the blue and red regions means where it indicates that oxygen is evolving more. So the more oxygen is evolved, the more aerobic bacteria are accumulated there. Okay. So here in his experiment, Engelmann experiment, most of the oxygen is released in blue and red regions. So that's the reason why the bacteria are accumulated at blue and red regions. Okay. They're accumulated at blue and red regions. Okay. You can see. So here when you come to this action spectrum and absorption spectrum. Okay. Action spectrum is a graph which shows effectiveness of photosynthesis at different wavelengths of light. So photosynthesis will take place in the visible range of light, that is 400 to 700 nanometers. The visible range of light is 400 to 700 nanometers of light. Okay. So where we can see, we can calculate the rate of photosynthesis in terms of oxygen produced. So in terms of oxygen produced at different wavelengths at time. Okay. So this is called action spectrum. So action spectrum is a graph which shows the effectiveness of wavelengths of light, that is the rate of photosynthesis at different wavelengths of light, which is not a direct measure. It is measured in terms of oxygen release. The more the oxygen release, the more the photosynthesis. So this is a measure to rate of photosynthesis. So you can see this is an action spectrum graph. Okay. Where you can see the rate of photosynthesis is at peak at these 2 points. Okay, where the absorbance of different wavelengths of light is at peak. So what is the significance of action spectrum? Okay. It is having a lot of importance, that is to establish the process, the rate of photosynthesis, at which pigment it is maximum. Photosynthesis occurs maximum in blue and red region of spectrum. Very least photosynthesis takes place in green and yellow light. Why? Because they are reflected back from the leaf. Okay. So what action spectrum shows is maximum photosynthesis will take place in blue and red region of light. Very little in green and yellow light. Okay. So this is about the action spectrum. So absorption spectrum means the absorbance of light by pigments at different wavelengths. So this is finally the proven by T.W. Engelmann. बाद में नेक्स्ट पर्सन है कि Cornelius Van Niel. What he has done, we'll see. He has conducted experiments on purple and green bacteria. He has conducted experiments on purple. Okay. And he has demonstrated that it is a light dependent reaction. And during this process, hydrogen is suitably oxidized and carbon dioxide is reduced to carbohydrates. So here you can see oxidation of water and reduction of carbon dioxide. So here you can see this is oxidized, this is reduced. Okay. So from carbon dioxide, carbohydrates are produced. Okay. So this is expressed 2Ha plus CO2 gives rise to 2A plus CH2O plus H2O. So this Cornelius Van Niel, so finally he has got into conclusion that is H2O, water, which is required for photosynthesis, is a hydrogen donor. It undergoes oxidized. So this H2O is split during light reaction, that is splitting of water takes place. And more specifically, it is called photolysis of water. So it is called photolysis of water. So photolysis means splitting of water in the presence of light. Okay, this takes place in plants. Okay. When you come to few bacteria, that is sulfur bacteria. So in sulfur bacteria, instead of water, H2S, hydrogen sulfide, is hydrogen donor. So on oxidation, you'll get sulfur. This was later proved by using radioactive isotope of oxygen. Okay. Later on it is proved by using a radioactive isotope. So here in sulfur bacteria, instead of oxygen, sulfur is released. Why? Because here H2S is hydrogen donor. Whereas In plants, water is hydrogen donor. Splitting of water. Here, splitting of hydrogen sulfide will take place. Okay. So, this is proved by taking radioactive isotope of oxygen. So, when you take the exact equation for photosynthesis in higher plants, the equation is 6CO2 plus 12H2O gives rise to 1 glucose molecule, 6 water molecules, 6 oxygen molecules. This is what finally Van Niel has demonstrated. That is, water is a hydrogen donor which undergoes splitting, that is photolysis of water. When you come to few bacteria like sulfur bacteria, इसमें hydrogen sulfide will split. So finally क्या आते हैं कि sulfur. Okay. So here the hydrogen donor is water in plants, whereas in sulfur bacteria it is H2S. Okay. So these are the early experiments which are done by many people. Like this, there are many other experiments also, like Mole's half-leaf experiment, which proves that carbon dioxide is required for photosynthesis. Okay, carbon dioxide is required for photosynthesis. You can test the presence or absence of starch by iodine test. Like what you have seen, Joseph Priestley, Van Ingenhouse, Julius Van Sachs, T.W. Engelmann, Van Niel, it all has given a conclusion about regarding each and every factor. Okay, we are studying about photosynthesis, the factors, the experiments which you have done, but where this photosynthesis will take place? Photosynthesis किधर होता है? The site of photosynthesis is chloroplast. So chloroplast is one of the largest cell organelle in plants, which is present only in plant cell. So chloroplast is the cell organelle in which photosynthesis takes place. So chloroplast में दो मेम्ब्रेन्स रहता, आउटर मेम्ब्रेन और इनर मेम्ब्रेन. इनर मेम्ब्रेन के नीचे स्ट्रोमा रहता, और इस स्ट्रोमा में थाइलेकोइड्स रहता, बहुत सारे थाइलेकोइड्स रहते. ए पाइल ऑफ थाइलेकोइड्स को ग्राना बोलते. पाइल ऑफ थाइलेकोइड्स को क्या बोलते? ग्राना. So inside grana, the light reaction takes place, and inside in stroma, the dark reaction takes place. Dark means light independent reaction. Light reaction means light dependent reaction. So grana, light reaction will take place. Stroma, dark reaction. And the membrane system is responsible for trapping light energy and for synthesis of ATP and NADPH. That is, during light reaction, light energy is converted to energy currency, that is ATP and NADPH. Okay. In stroma, by using this ATP and NADPH, carbon dioxide is reduced and it will form starch. So carbon dioxide से कहा जाता है कि starch आता है. So this is site of photosynthesis. The site of photosynthesis is chloroplast. So आप क्या देख रहे कि diagram of chloroplast. इसमें आप देख सकते हैं. This is outer membrane, and the next is inner membrane. outer inner membrane के बीच में स्पेस को क्या बोलते कि intermembrane space और इनर मेम्ब्रेन के नीचे मैट्रिक्स. सो मैट्रिक्स को क्या बोलते? स्ट्रोमा. स्ट्रोमा में सैक लाइक स्ट्रक्चर्स, इसको क्या बोलते? थाइलेकॉइड्स. इसका नाम थाइलेकॉइड्स. एक ग्रुप ऑफ थाइलेकॉइड्स, ए पाइल ऑफ थाइलेकॉइड्स, इसका नाम ग्रैनम. ग्रैनम इज सिंगुलर, ग्रैना इज प्लूरल. Grana or grana are connected by stroma lamellae. And inside thylakoid there is a space that is called lumen. और स्ट्रोमा में थाइलेकॉइड्स ग्राना के साथ क्या रहता है कि डीएनए और राइबोसोम्स, 70s राइबोसोम्स भी रहता. सो क्लोरोप्लास्ट इज ए सेल्फ रेप्लिकेटिंग सेल ऑर्गेनल. इसमें डीएनए रहता है और राइबोसोम्स भी रहता. सो इसलिए इसको क्या बोलते हैं कि इट इज सेल्फ डुप्लीकेटिंग सेल ऑर्गेनल लाइक माइटोकॉन्ड्रिया. सो स्ट्रोमा में डार्क रिएक्शन होता है और ग्राना में लाइट रिएक्शन. So light reaction takes place in granum and dark reaction takes place in stroma. Okay. So when you come to inside thylakoid, thylakoid के अंदर क्या रहता? So thylakoid के अंदर क्या रहता है कि photosynthetic pigments रहता. The major one that is chlorophyll. So what you are seeing, this is structure of chlorophyll. A chlorophyll में major pigment is chlorophyll A. So major photosynthetic pigment is chlorophyll A. So this is having chlorophyll A structure. If you take, it has a ring that is called porphyrin-like ring-like structure, and a tile that is hydrocarbon tile. When you come to chlorophyll A and chlorophyll B, both are more or less same. Only the one difference is the group. The methyl group in chlorophyll A, instead of methyl group here, it is a CHO in chlorophyll B. Remaining all same. In the porphyrin head, there is magnesium which is surrounded by nitrogen. Okay. So this is the structure of chlorophyll molecule. A chlorophyll molecule में ring porphyrin ring रहता और hydrocarbon tile. A porphyrin ring में center में magnesium रहता surrounded by nitrogen. So chlorophyll A is major photosynthetic pigment, and remaining all pigments are called accessory pigments. So chlorophyll A is the major photosynthetic pigment which plays an important role in photosynthesis. So here you can see the main pigment of photosynthesis, chlorophyll A. और बाकी पिगमेंट्स को क्या बोलते हैं कि एक्सेसरी पिगमेंट्स. सो एक्सेसरी का मीनिंग क्या है? Accessory means they are not directly involved in photosynthesis, but they help photosynthesis. They absorb wide range of wavelengths of light and transfer to chlorophyll A. So chlorophyll A will play a major role. Accessory pigments will transfer the wavelength of light to the chlorophyll A. So chlorophyll B, xanthophylls, carotenoids, उसको क्या बोलते हैं कि accessory pigments. The main pigment, accessory pigment. Main pigment का नाम chlorophyll A. Accessory pigments क्या है कि chlorophyll B, xanthophylls, carotenoids. So we can extract these pigments by using paper chromatography, a simple experiment. A paper chromatography we can take any part of the plant. That is, for example, if you take leaves, grind the leaves properly in a mortar and pestle. Take a paper, chromatographic paper, keep a dot on this. Okay. Put this chromatographic paper in a solution. Okay. And you can see from this pigmented dot, we can see different pigments move depending upon the molecular weight they reach. The low molecular weight will be at the bottom. The high molecular, next lower will be chlorophyll A. The highest will be chlorophyll B, A, carotenoids, xanthophylls. We can see that. Okay. So here, if we take chlorophyll pigments, the photosynthetic pigments, the first one is chlorophyll A, which is bright or blue-green chromatography. Chlorophyll B, yellow-green. Xanthophylls, which are yellow in color. Carotenoids, yellow to orange, which are present in carrot. Okay. And these photosynthetic pigments can be seen in a paper chromatography experiment with the colors. Chlorophyll A, blue-green. Chlorophyll B, yellow-green. Xanthophylls, yellow. Carotenoids, yellow to orange. These are different pigments you can see. The bands of pigments you can see. So here you can see. This is a chromatography paper which is kept in a solvent, where a dot of original pigment solution is kept. So when it is kept in a solvent, along with solvent, we can see the pigments will move. You can see different bands of pigments. Like you can see, that is chlorophyll B. The bottom is chlorophyll B. Blue-green, chlorophyll A, xanthophylls, carotenoid. These are different bands of pigments. This is the use of this experiment, that is the what we call the paper chromatography. Okay. So अभी तक हमें पढ़ाया the experiments, और बाद में हमें क्या पढ़ाया कि the chloroplast structure of photosynthetic pigments. Okay. And these photosynthetic pigments किधर रहता? ये thylakoid के अंदर रहता. They're present inside the thylakoid. And these pigments are organized into photosystems. They're arranged systematically. So this photosystems, like there is, based on the arrangement, there is photosystem 1 and photosystem 2. In short, we'll call them as PS1 and PS2. In short, इसको क्या बोलते हैं कि PS1 and PS2. इसमें आप देख सकते हैं लाइट हार्वेस्टिंग कॉम्प्लेक्सेस, इसको क्या बोलते हैं कि photosystems. A light harvesting complex में क्या रहता है? इसमें बहुत सारे प्रोटीन्स और पिगमेंट मॉलिक्यूल्स रहते हैं. इसमें बहुत सारे प्रोटीन्स और पिगमेंट मॉलिक्यूल्स रहते हैं. So this light harvesting complex means which will take the light. So इसमें PS1 और PS2. Okay. So PS1 में reaction center, that is which has chlorophyll A, उसको क्या बोलते हैं कि P700. So PS2 को क्या बोलते हैं कि P680. P680. क्लोरोफिल A के साथ क्लोरोफिल B, कैरोटेनॉइड्स और xanthophylls भी रहते हैं. So in photosystem there is an antennae and reaction center. Antennae will receive the light and it will be sent to reaction center where the further process will take place. That is the excitation of chlorophyll. This all will take place in reaction center. So PS1 का reaction center का नाम P700 और PS2 का reaction center का नाम P680. देख रहे फोटोसिंथेसिस में बेसिकली दो stages रहता. One is light reaction. It is called light dependent reaction. Dark reaction, which is light independent reaction. What is this light? What is this dark reaction? So light is a photochemical phase. Photo means which is dependent on light. It is a biosynthetic phase where we can see only changes in the cell in the absence of light. A light reaction किधर होता? thylakoids grana में होता. Dark reaction किधर होता? stroma में होता. It is dependent on light directly. So it is light dependent reaction. It is independent of light. It is catalyzed by many enzymes. So इधर क्या होता है कि light absorption, photolysis of water, that is splitting of water, oxygen भी release होता है और finally you'll get energy currency, that is ATP and NADPH. So in light reaction, the light energy is converted to ATP and NADPH. ATP and NADPH, they are utilized in dark reaction for reduction of carbon dioxide to form carbohydrates. So you can come to one conclusion here. Dark reaction is dependent on light reaction. The end products of light reaction, that is ATP and NADPH, are used in dark reaction for synthesis of carbohydrates. These are the two stages of photosynthesis where the light and dark reaction. So पहले हम देखेंगे लाइट रिएक्शन में क्या होता. So whatever the energy present in light, it is called photons. So whenever light falls on chlorophyll A, so what happens is you can see the excitation of chlorophyll A. So here the excitation means the electrons which are present at ground level, they are moved to higher levels and they come out of that one. That means electrons come out of the chlorophyll A molecule, which is called excitation. इलेक्ट्रॉन्स को क्या होता है? इलेक्ट्रॉन्स क्लोरोफिल ए से बाहर आए, इसको क्या होता है? So these electrons are accepted by electron carriers. So पहले इलेक्ट्रॉन कैरियर को क्या बोलते हैं कि प्राइमरी एक्सेप्टर. And these electrons which are released, these are high energy electrons. Electrons में high energy रहता. And these high energy electrons, they move through different electron carriers. A picture में क्या दिख रहे कि these electrons which move through different electron carriers again come to the same starting point. That is, FD means ferredoxin. plastoquinone, cytochrome complex, plastocyanin, a electron carriers. And through these electron carriers, the electrons will reach to again this place. When they come to the same starting point, it is called cyclic electron transport. So इसको क्या बोलते हैं कि cyclic electron transport. इसमें अब देख रहे electrons are moving in a cyclic form. ऐसे साइक्लिक फॉर्म में मूव होने इलेक्ट्रॉन्स को एक प्रोसेस को क्या बोलते हैं कि साइक्लिक इलेक्ट्रॉन ट्रांसपोर्ट. सो इन दिस साइक्लिक इलेक्ट्रॉन ट्रांसपोर्ट इसके साथ क्या होता है कि साइक्लिक फोटोफॉस्फोरिलेशन. What is meant by photophosphorylation? Phosphorylation means addition of phosphate to ADP, then we will get ATP in the presence of light. Okay. इसमें PS1 is participating, that is the reaction center का नाम P700. So electrons are moving within the photosystem. Okay. So इधर क्या होता है कि that is photophosphorylation happens in stromolamellae. Because as it lacks PS2 and NADP. Okay. But whereas when you come to granulamellae has both PS1 and PS2. The excited electrons comes back to the PS1. Again it reaches to the PS1 itself. इसमें सिर्फ ATP synthesize होता है, no NADPH2. Okay. Only occurs beyond 680 nanometers. A cyclic photophosphorylation में हमें end products लिया तो सिर्फ ATP is released. इसमें सिर्फ ATP release होता है. Oxygen or NADPH2 is not released. सिर्फ ATP is released and it occurs in stroma lamellae. And here the electrons will flow in cyclic manner. They will start from PS1 and again they reach to PS1. So this is the formation of ATP during cyclic electron transport. वो process को क्या बोलते हैं कि cyclic photophosphorylation. Okay. So in this, the electrons are moving in a cyclic pattern and by the end we will get product is ATP. ऐसे हमको रहता नॉन साइक्लिक इलेक्ट्रॉन ट्रांसपोर्ट. इसमें electrons will move in a non-cyclic pattern where they'll start and they will end with another substance. इसको क्या बोलते हैं कि non-cyclic. इसमें non-cyclic photophosphorylation रहता और इसमें ATP and ADPH2 ऑक्सीजन रिलीज होता है. So this is about the cyclic photophosphorylation and the cyclic electron transport. So cyclic and non-cyclic will take place simultaneously. Okay. And here you should know the important aspects in cyclic electron transport. Here only PS1 is involved. Only ATP is formed. No oxygen, no NADPH2. And it occurs in stroma lamellae. And it occurs beyond 680 to 700 nanometers. Okay. So this is what happens in cyclic electron transport, which is associated with cyclic photophosphorylation. Finally, we get ATP. So till now we have learnt about the experiments, the site of photosynthesis, the chloroplast structure or parts, stages of photosynthesis, that is light reaction, dark reaction, light or dark reaction का differences, light reaction में क्या form होता है, end products क्या है, dark reaction के end products क्या है. Dark reaction is dependent on light reaction and products. और लाइट रिएक्शन में क्या होता, इलेक्ट्रॉन ट्रांसपोर्ट और फोटोफॉस्फोरिलेशन। इलेक्ट्रॉन ट्रांसपोर्ट में साइक्लिक रहता, नॉन साइक्लिक रहता। फोटोफॉस्फोरिलेशन में भी साइक्लिक और नॉन साइक्लिक फोटोफॉस्फोरिलेशन। Finally, what we'll get? ATP, NADPH2 and oxygen. Oxygen is the byproduct. The main product is ATP and NADPH2. So this is are the differences between the light and dark reaction. I hope you all understood till now what we discussed. So let us meet in the next session. फिर मिलेंगे in the next session. Thank you all. We'll meet in the next session. Follow SPSVERSITY on the social media.